Assalamualaikum and hi everyone. Today we are going to learn about drug toxicity and adverse reaction. In this topic, we are going to learn about dose response, pharmacokinetics, and toxicokinetics, the drug toxicity testing in animal study, the safety pharmacology and clinical trials, adverse drug reactions, and also principles of treatment of poisoning. Let's start with the dose response. The dose response relationship is the most basic and the most important fundamental concept in the field of toxicology because there are so many health and economic assessments as well as regulatory decisions often depend on the integrity of this relationship. In a true dose response relationship, there is some measurable effect that is proportional to the amount of the drugs received. Uh, Three general assumptions that must be considered when evaluating the dose response relationship, which are the drugs interact with the molecular or receptor sites to produce a response. And the second one is the production of the response or the degree of response is correlated to the concentration of the drug at that receptor site. And the last one is the concentration of the drug at the receptor site is related to the dose of the drug received. Credit response on the y-axis, the response is expressed as the percentage of maximal response plotted as a function of the concentration of drug A, which represent at the receptor x-axis, and the hyperbolic shape of the curve in this panel becomes sigmoid when plotted semi-logarithm as in the panel B. The concentration of drugs that produces 50% of the maximal response quantifies drug activity and it is referred to as the EC50, effective concentration of agonist for 50% response. Quantum dose response relationship, the percentage of the population affected increased as the dose is increased. Then the relationship is quantal is that the effect is judged to be either present or absent in a given individual. And this quantum dose response phenomenon is used to determine the LD50 of drugs. A quantum dose response curve for the therapeutic effects of a drug to generate ED50, which is the concentration of a drug at which 50% of the population will have the desired response, and a quantum dose response curve for lethality by the same agent. While when we talk about therapeutic index or TI, it is very widely, the values is very widely, starting from 1 to 2 to more than 100. So the drugs with a low therapeutic index must be administered with caution. For example, the cardiac glycosadigoxin and cancer chemotherapeutic agents. While the agents with a very high therapeutic index, for example, penicillin, are extremely safe in the absence of known allergic response in a given patient. Let's look at to the pharmacokinetics versus toxicokinetics. Right, before we go further, let us recap about ADME. So basically, ADME is the fourth step of pharmacokinetics and the abbreviation for absorption, distribution, metabolism, and also excretion. When we talk about absorption, it actually describes how a chemical or drugs enters the body, and it relates to the movements of a drug from the administration sites to the bloodstream. The second is distribution, right? So just imagine once a drug has been absorbed, it moves from the absorption site to the tissues around the body, and the distribution from one part of the body to another is typically accomplished via the bloodstream, but it also can occur from cell to cell. The third one is metabolism. When we talk about drug metabolism, it is the biotransformation of a drug by organs or tissue, and most importantly, including the liver, kidney, skin, or digestive tract, so that the drug can be excreted. To facilitate removal via feces or urine, the drug compound is altered to become more water soluble. The drug metabolism can result in toxicity, for instance, by creating damaging bioproducts or a toxic metabolite. And the last one is about excretion, and this is the last step of ADME. All right, so why ADME is important? So basically, when we talk about drug discovery and development, all the researchers must examine the activity of a drug in the body to assess the safety and also the toxicity of that drug. And when we talk about drug metabolism and pharmacokinetic study, such as ADME and toxicology studies, both are critical steps in this process. And the data collected would be used in by the researchers if a drug is viable and can provide specific target for future research and development. Okay, when we talk about the alterations in ADME, it will focus on the drug absorption and also the drug elimination. For drug absorption, aspirin poisoning is a leading cause of overdose morbidity and mortality as reported to US poison control centers. And in therapeutic dosing, aspirin reaches a peak plasma concentration in about 1 hour. However, aspirin overdose may cause spasm of the pyloric valve, delaying entry of the drug into the small intestine. Aspirin, especially enteric coated forms, may coalesce into basophils, reducing the effective surface area for absorption. And the peak plasma salicylate concentrations from aspirin overdose may not be reached for 4 to 35 hours after ingestion. And about the drug elimination, let's focus on the table here. And this table actually lists some pharmaceutical notorious for their predilection to have initial symptoms develop after a typical 4 to 6 hour emergency medical observation period. In therapeutics, a drug typically produces numerous effects, but usually only one is sought as the primary goal of treatment. And the most of the other effects are undesirable effects for that therapeutic indication. The side effects of drugs usually are bothersome but not deleterious. Other undesirable effects may be characterized as toxic effects. For dose-dependent reactions, toxic, the effects of drugs may be classified as pharmacological, pathological, or genotoxic. Typically, the incidence and seriousness of the toxicity is proportionately related to the concentration of the drug or the compound in the body and to the duration of the exposure. Pharmacologic toxicity, for example, the CNS depression produced by barbiturates is largely predictable in a dose-dependent situation. The progression of clinical effects goes from anxiolysis to sedation to somnolence to coma. Pathological toxicity. For example, acetaminophen is metabolized to non-toxic glucuronide and sulfate conjugates and to a highly reactive metabolite NAPQI, Q1, sorry, via CYP isoforms. At a therapeutic dose of acetaminophen, NAPQ1 binds to neophilic glutathione, but in acetaminophen overdose, the situation of overdose, glutathione depletion may lead to the pathological finding of hepatic necrosis due to shunting of NAPQ1 toward interaction with nucleophilic cellular macromolecules. How about genotoxic effects? So, for example, ionizing radiation and many environmental chemicals ataupun drugs are known to injure DNA and may lead to mutagenic or carcinogenic toxicities. And many of the cancer chemotherapeutic agents may be genotoxic. Let's move to the second, which is allergic reactions. When we talk about allergy, it is an adverse reaction mediated by the immune system, which resulted from previous sensitization to a particular chemical or compound or to one that is structurally similar. Allergic responses have been divided into four general categories based on the mechanism of immunological involvement. The third one is idiosyncratic reactions and pharmacogenetic contributions. Basically, idiosyncrasy is an abnormal reactivity to a chemical that is peculiar to a given individual, and the idiosyncratic response may be extreme sensitivity to low doses or extreme insensitivity to high doses of drugs. A common mechanism is covalent drug binding to serum proteins that leads to the presentation of a foreign heptide, resulting in an immunotoxicological response. When we talk about drug-drug interaction, we can say that the interaction can occur during the absorption, protein binding, metabolism, receptor binding, and also therapeutic action. Okay, now let's move to drug toxicity testing in animal. There are two main principles of assumptions underlie all descriptive toxicity tests performed in animals. First, those effects of chemicals or compound produced in laboratory animals, when properly qualified, apply to human toxicity. When calculated on the basis of dose per unit of body surface, toxic effects in human being usually are encountered in the same range of concentrations as those in experimental animals. On the basis of body weight, human beings generally are more vulnerable rather than experimental animals. Normally, OECD guideline or OECD test guidelines, in short, TGs, is used for the tasting chemicals or compound that are collected of the most relevant internationally agreed tasting methods. So it actually the details of the procedures for regulatory safety testing, chemical notification and registration. And another component of OECD teaches is the mutual acceptance of data, which then helps to address governmental requirements and provide a basic for cooperation among national authorities. Right. So usually toxicity testing is characterized by the exposure duration. For example, acute toxicity study is conducted to determine the short-term adverse effects of a drug when administered in a single dose or in multiple doses during a period of 24 hours in two mammalian species. The test animals, typically rodents, for example, rats or mice, are observed for a period of several days to 2 weeks after dosing, and the observations of deviant behavior, growth, or mortality are recorded. Historically, the primary focus of an acute toxicity test was to determine a chemical's median lethal dose, or LD50, which is the dose that causes death in 50% of the test animals. The second one is the subchronic toxicity studies. Actually, the adverse effects occurring after the repeated of or continuous administration of a test sample for up to 90 days or not exceeding 10% of the animal's lifespan. They provide information on target organ toxicity and the bioaccumulation potential and are designed to determine no observed adverse effects levels, or sometimes you can see as in short NOELs, which are used to establish standards or guidelines for human exposure. This subchronic study is also not designed to assess effects that have a long latency period, such as in cancer, but do provide information that can be used in setting doses for chronic toxicity and carcinogenicity studies. And the exposure durations for subchronic studies are typically 28 or 90 days. Chronic toxicity study, and the objective of this type of study is to characterize the profile of a substance or chemicals in mammalian species, normally rodents again. Animals per sex per group should normally be used at each dose level, while for non-rodents, a minimum of 4 per gender or per sex per group is recommended. At least 3 dose levels should be used in addition to the concurrent control group, and the frequency of exposure normally is daily but may vary according to the route chosen. Either it's oral, dermal, or inhalation, and should be adjusted according to the toxicokinetic profile of the test substance or compound. The duration of the exposure period should be 12 months, and the study report should include the measurements, for example, the weight, and the regular detailed observation, such as hematological examination, urinalysis, clinical chemistry, and as well as necropsy procedures and histopathology. Now let's move to the fourth learning outlines, which is safety, pharmacology, and clinical trials. Clinical trials of drugs are designed to acquire information about the pharmacokinetic and pharmacodynamic properties of a candidate drug in humans. The efficacy must be proven and an adequate margin of safety established for a drug to be approved for sale. The FDA regulated clinical trials typically are conducted in 4 phases. In such a phases 1 until 3 are designed to establish safety and efficacy, while phase 4 include the post-marketing trials, delineate additional information regarding new indications, the risk, and optimal doses and schedules. This figure and the table in the next slide summarize the important features of each phase of clinical trials. Please note the attrition at each successive stage over a relatively long and costly process. When initial phase 3 trials are complete, the sponsor, usually the pharmaceutical company, applies to the FDA for approval to market the drug. Testing in humans begins only after sufficient acute and subacute animal toxicity studies, as per mentioned previously. So, chronic safety testing in animals, including carcinogenicity studies, is usually done concurrently with the clinical trials. The accumulating and analyzing all necessary data often requires between 4 to 6 years of clinical testing. In each of the 3 formal phases of clinical trials, the volunteers or patients must be informed of the investigational status of the drugs as well as the possible risks, and must be allowed to decline or to consent to participate and receive the drug. And these regulations are based on the ethical principles set forth in the Declaration of Helsinki.